So this is a video on glycolysis, basically carbohydrate metabolism. We're also going over the electron transport chain, and we're also going over the anaerobic and aerobic respiration that goes on with the citric acid cycle. So I have a picture here, and this picture is basically going over what happens to C6H12O6, or rather glucose. If you go over basic biochemistry reactions in the body, the idea of dehydration synthesis and hydrolysis, you see that the ATP is kind of hydrolysizing itself into ADP and a phosphate group. That phosphate group, going from glucose to glucose 6-phosphate, is being added to that carbon chain. Then, because I think it's the idea of an isomer, they can have the same C6H12O6, but their structure is a little bit different. I think that's why it can become fructose 6-phosphate. From fructose 6-phosphate, we have another hydrolysis of ATP, adding that phosphate group. Now having two phosphate groups known as fructose 1,6-diphosphate. And then you have the cleavage period, which is just you're breaking that fructose 1,6-diphosphate molecule into two separate pieces. And then you have oxidation going on with the niacin-derived cofactor. We have the riboflavin-derived cofactor, which essentially works in the citric acid cycle. Cofactors are very important. And then you have dephosphorylation. So you have from the chain, right after the point of cleavage, two more phosphate groups that are added. And essentially, you have the production of 2-pyruvic acid through dephosphorylation. Now you have anaerobic fermentation and aerobic respiration. What's important to understand is anaerobic fermentation is just the production of lactic acid. Aerobic respiration continues with the citric acid cycle. So at the end of glycolysis, you had 2-pyruvic acid. All right, these 2-pyruvic acid are now being released into CO2. Now it's a 2-carbon structure. They're known as acetyl groups, C2. You have coenzyme A. This makes acetocoenzyme A. It has circular function, and this mixes with oxaloacetic acid, a 4-carbon structure molecule. That forms a 6-carbon structure molecule. It's rearranged, and then you lose the CO2. You have a niacin-derived cofactor that goes on. It becomes a 5-carbon structure. From this 5-carbon structure, you have another niacin-derived cofactor. From this niacin-derived cofactor, you have a release of the CO2. So now you have 4-carbon structure, and then you have the production of ATP through GTP as well. Then you have a riboflavin-derived cofactor. You lose some H2O, and it becomes oxaloacetic acid, so it's a cyclical cycle. The next topic that I want to go over are matrix reactions and the membrane reactions. So what's important to understand here is that this is happening within the mitochondria. So I have a picture that has flavin mononucleotide, iron sulfur centers, coenzyme Q, copper ions, and cytochromes. This picture is essentially showing what is happening basically along the membrane reactions. It's just kind of like a cascade in a way. The final electron acceptor of the electron transport chain is oxygen, and we see that this oxygen goes to make water, how the body, I guess, produces water. And we can take a look at what happens at the actual phospholipid bilayer with the different enzymes or proteins at the end of this, at the end of the electron, the ETC. So we're seeing that as well. So the goal is to make water, because we know the byproducts of glycolysis, overall glycolysis with electron transport chain, and all this type of metabolism is CO2 and H2O.